everybody! This is our second video. It is going to be all about the mitochondria and the structures of the mitochondria, how we transport things into the mitochondria, out of the mitochondria, and throughout the mitochondria. So we're just going to be really looking in a lot of depth at the mitochondria. So let's just think of a mitochondrion. That's, a mitochondrium is the singular for mitochondria, which is the plural. And we're going to have this organelle that is membrane bound, or membrane covered. It has this outer membrane, which we have circled here. This is the outer membrane that just surrounds the entire mitochondrium. This is a membrane that is about half lipid, half protein. It's double layered. And because of this, things can't just diffuse across it, just like your regular cellular membrane. Not everything can just diffuse across your, normal cellular membrane around the outside of the entire cell. And it's the same situation for our outer membrane in the mitochondria. If we want to move things, substances, across this membrane, we're going to need to have various transport mechanisms in the form of carrier proteins, just like we have in the regular old cell membrane. There are proteins that carry sodium ions across the cell membrane. And here we'll be looking at carrying things that are important for what the mitochondria is doing. And the mitochondria is essentially taking pyruvate, or a metabolite of pyruvate, which is acetyl-CoA. That might sound familiar to you. If not, that's fine. It will eventually, soon. By our next video, acetyl-CoA will be very familiar to you. But acetyl-CoA is something that goes into the Krebs cycle, and it can't just, come straight from pyruvate, which came from glycolysis, and just bam, its way right into the mitochondria and the Krebs cycle. There has to be different mechanisms of carrying it across. Another thing that we need to transport across is NAD or NADH. This also can't just cross the membrane. It needs to be transported, and it's going to be converted into other intermediary sorts of substances that can cross the membrane through various carrier proteins, various transport mechanisms, and then be reconverted back into NAD and NADH. It's kind of interesting. You start out with NAD or NADH out here, you turn it in the cytoplasm, is where I'm pointing to, and you turn it into something else. That something else can cross that outer membrane and then get reconstituted into NAD and NADH. So it's, change it, move it, change it back again. So that's what we're going to be looking at as we look at this outer membrane. Now there's also an inner mitochondrial membrane. That's this all in here that sort of looks worm-like a little bit in there. But it's just another membrane, and again, as a membrane, things aren't just able to pass however they want across them. We need to have transport mechanisms and carrier proteins for that too, in order to get things into or out of the inside. So if you see here, they've kind of cut a little piece of that inner membrane, and you can see what's inside, which is the matrix, so like a space in there. And that is where, inside the matrix, is where the Krebs cycle is happening, the reactions that are creating the energy, the electron transport chain is happening. Now, within the matrix, as part of the inner membrane, but on the inside of that inner membrane, you can see how it's all ridged. That inner membrane is just, folded and ridged. Those ridges are called cristae. So here it says crista, it's pointing at just one ridge, but the plural for that would be cristae. And embedded on those are the proteins for the electron transport chains. So here it has that labeled as enzyme complexes. Those are proteins that act as enzymes that are bundled together to form the electron transport chain. And they are in that matrix, stuck on the folds, on the cristae. And they are going to be obviously super important for making energy. Now, that energy, once it's made in the matrix of the mitochondria, it needs to then cross the inner membrane, It will go into the space between the inner membrane and the outer membrane, which is called the intermembrane space. And then it needs to also cross the outer membrane. So we have various transport mechanisms to cross things like ATP as it's moving from inside the mitochondria across the inner membrane into the intermembrane space, and then across the outer membrane out into the cytoplasm. Just like we have transport mechanisms to take metabolites, things like acetyl-CoA or NAD, NADH, move it from the cytoplasm into or across the outer membrane into the intermembranous space, then across the inner membrane into the matrix, where we can then use, say, NAD and NADH in the electron transport chain. So we're going to look at some of those transport mechanisms now. Or in a second. Our next slide is just going into some detail that I just talked about the cell membrane, or the outer membrane structure, so the outer mitochondrial membrane structure. About half protein, half lipid. It's keeping the inside of the mitochondrium separate from the cytoplasm, so it's just an enclosed entity inside of the mitochondria. It is not just continuous with the cytoplasm. It's creating a different space, or a compartmentalized space, that is the inside of the mitochondria versus the rest of the cell. And across the outer membrane is where we're going to be moving our metabolites like acetyl-CoA and NADH into the, and there's a typo on this slide, it should say intermembrane space, not intramembranous space, but the intermembrane space, so between the outer membrane and the inner membrane. Now, the transport mechanisms, here we can get into them. They're called porins, and this mouthful, which is the translocase of the outer membrane complex, or the TOM. So we can just call it TOM. Let's just call it TOM, make life a lot easier. So we're going to be looking at the porin, and we'll be looking at TOM. Okay, so what is a porin? Let's up a little bit. So a porin is a voltage-dependent anion channel. Voltage-dependent refers to that whether it is open or closed depends on the voltage of that outer membrane. So if you think back to transmembrane potential of the actual, cell membrane around the entire cell, that voltage changes in relation to what the fluid is, or what the voltage is outside of the cell. So typically the cell would be negative inside and positive outside. This is a similar sort of situation where you have a membrane separating two different places, the cytoplasm of the cell versus what's inside the mitochondrion, and there will be an electrical charge on that membrane, just like there is on the outside of the cell itself. So whatever the voltage is on the mitochondria at any given time is going to determine whether the porin is open or closed. An anion is a negative ion. A cation is a positive ion, so, negative, positive. So this is an anion channel, which would make you think, okay, anions move through it, and yes, they do when the porin is open. But cations can move across it when it's closed, which makes it kind of confusing and makes it go, okay, why do they call it an anion channel if cations can also cross it? But just because the open state allows the anions to move. So when we're thinking of the porin being open, we're talking about moving the anions across. We're talking about moving things like ATP across. ATP obviously is made inside the mitochondria, and it needs to get out of the mitochondria, so it can move out using an open porin. In the closed state, we have the porin being permeable to cations, so positive ions instead. So things like calcium, especially calcium, is important here. And why I say especially calcium is that calcium is very, very important as a cofactor. So what is a cofactor? A cofactor is a molecule. Could be a compound, but typically just a molecule that enables a enzyme to work. I'm going to change pens. That pen is not working so good. So without our cofactor, our enzyme won't work in a lot of cases. So cofactors are very common things that enzymes use, and it helps the enzyme to work better, allows it to work at all. And there is one specific enzyme, pyruvate dehydrogenase. So there's that ACE at the end, which tells you that this molecule is an enzyme. It says pyruvate, so you know it's going to have something to do with pyruvate, which came from glycolysis. This is an enzyme that you should remember. I know I said we don't memorize metabolic pathways here, but pyruvate dehydrogenase is something that we are going to just see a lot of, because it's one of those kind of crossroads points in the metabolic pathways where we go one way or we go another, and a lot of it has to do with whether we have pyruvate dehydrogenase available or not. And pyruvate dehydrogenase needs calcium as a cofactor in order to work. So if we don't have pyruvate dehydrogenase, we're unable to take the pyruvate, turn it into, say, acetyl-CoA, which is what can enter into the Krebs cycle. We're not able to do that without pyruvate dehydrogenase. So without calcium, we can't do that. We can't have pyruvate dehydrogenase working. So calcium is really important to be able to get into the mitochondria. So for this porin to be closed is actually essential for the pyruvate dehydrogenase to work and for the Krebs cycle to actually proceed and create ATP. Now in the closed situation though, it's going to be impermeable to ATP, so that ATP would then, if it stayed closed, be stuck in the mitochondria, but it will open again when the voltage changes, as we create more energy. We won't keep doing that influx of calcium, but that's okay because we got the energy that we needed. So what closes it? High voltage. So this is about 50 millivolts. It can be anywhere around 30. They'll start to close. At 50, it'll be really closed. So all the porins would be closed. So a super high voltage, in other words, is going to close the porin and allow the calcium in. Or there are times when a chemical like NADH can block the porin. So when you have NADH building up, think about the situation that causes you to have NADH building up. NADH is used in the electron transport chain. And so you're in the middle of the Krebs cycle, and if you would be using the Krebs cycle, you'd be taking the electrons off of NADH and you'd be bouncing them along the electron transport chain, and the end result, or the end electron acceptor, would be oxygen. And that whole process of the electrons going from NADH into oxygen, is going to give us energy. So ATP. That is the electron transport chain really, really quick. So NADH, oxygen, ATP. Now, if you don't have oxygen, well, you can't do that. You're not making ATP. This NADH is just stuck. So if you're anoxic, which means no oxygen, so anoxia, here, your NADH builds up because it's got no final electron acceptor here. Calcium comes in, which is a good thing. It can encourage pyruvate dehydrogenase, which is what you need to start the Krebs cycle in the first place. But too much calcium can cause apoptosis, starting from the mitochondria. So when you allow a ton of calcium, too much calcium into the mitochondria, it can actually kill the cell. And this is obviously a big problem. That's why you don't want your cells to be anoxic for too long, or to not have enough oxygen to run the electron transport chain. Not only do you not get ATP, but you also can be actually killing your cells. But normally, these are just sort of a nice little balance here. The porin opens, you get some, ATP out, anions can come in, it closes, calcium comes in, activates the pyruvate dehydrogenase, you can start the Krebs cycle, you do have oxygen normally, and you get your ATP out of it and everything works great. Now the porin is also really highly integrated, or really dependent on, involved with the translocase of the outer membrane, or the TOM. So remember we talked about the TOM. And here is the TOM. So over here we have, let's use a different color. Here's TOM. This is our outer membrane, so here is the TOM, because that's part of the outer membrane. On here, we also have the inner membrane, and on that inner membrane, we have the translocase of the inner membrane. That's TIM. So translocase, inner membrane, TIM. So TOM and TIM. These are important for carrying other metabolites across the outer membrane in the case of TOM, and the inner membrane in the case of TIM. Now, the porin helps to position the TOM22. So if you see how there's all these different little proteins all clustered around, and they're all named TOM with a number. Those are all the proteins that contribute to creating the TOM protein carrier, the TOM transport mechanism here. So the TOM22 is really involved with the porin. It is positioned by the porin, activated by the porin. Without the porin, TOM22 can't really do its job, doesn't do its job. The same thing is true of the TIM, where you have all these different little TIM proteins. They even have small TIMs, which is kind of funny, but all these little TIM proteins with the numbers, and the TIM22 is made of porin, or is made by porin, I should say, positioned by porin, not made of porin. But porin helps to activate the TIM22 and allow it to do its job as part of the TIM. In turn, others of the TOM proteins and TIM proteins help to create the porin in the first place. It would be the TOM20, where's the TOM20? Right there. And the TOM40 is also, so there's the TOM40. They help to create the porin, as do the TIM20 and the TIM40, which you don't need to, they're not even on there. So anyway, the point of this is, so you don't need to, you're not going to get like a diagram where you have to point out which TIM or TOM is here. Just to know that they are made of a bunch of different proteins, that the porins and the TOM and the TIM are dependent upon each other, that the porin helps to create the TOM and the TOM helps to create the porin. They're all super integrated, and they exchange, or they move different metabolites. So TOM and TIM will move one kind of metabolite, and the porin will move a different kind of metabolite. So they're all very important for moving things across the mitochondrial membranes. Okay, so we've already talked about this too, so I'm getting a little bit ahead of myself, but then we'll just reiterate it quickly here. So we know that the inner membrane has those folds, and we call those folds cristae. Inside the inner membrane is the matrix, and we have, the TIM. So we have the TIM that helps to carry things across the inner membrane. And we also have something else called the dicarboxylate carrier. The dicarboxylate carrier is important for carrying things important for the Krebs cycle across that inner cell membrane. So that's just another sort of carrier that allows things to move across those cell membranes, or the mitochondrial membranes. So far we've seen, as transport mechanisms, we've seen porins, we've seen the TOM, we've seen the TIM, and we have our dicarboxylate carrier, and all of them together, all four of these are transport mechanisms across mitochondrial membranes. Okay, so all are important for their own thing. Everything, all the different intermediary products that are going into the Krebs cycle or that are involved with the Krebs cycle or with the electron transport chain or just ATP. They're all going to have their own way of moving across those membranes, and will be one of these four. Okay, now here's just a recap of the translocase of the outer membrane, the TOM. So it's actually linked to the TIM, so it's essentially like a double door, if you think of it. If you're trying to get into or out of the mitochondrial matrix, you have to go, if you're coming from the cytoplasm, you have to go through TOM and then through TIM. If you're coming from the matrix and trying to move out into the cytoplasm, then you're going to have to go through TIM and then through TOM. We saw that it had a bunch of proteins on the TOM. They're all called TOM proteins. So that makes life easy. So we have just a bunch of proteins that are contributing to this carrier. Now, same for the translocase of the inner membrane, the TIM, linked to the TOM. So we know that. Again, lots of proteins. It's very similar to TOM in that it has the proteins, just they're called TIM proteins instead of TOM proteins. Okay, so enough about the membranes. Let's look at the matrix. So this is what's happening inside, or what we have inside. So here you can kind of see from a different view. This would be the outer membrane of the mitochondria. Here's the intermembrane space. Here we have the inner membrane. You see the cristae all folded. And then inside we have the matrix, and those white and kind of purple spots, those, and kind of greenish over here too, those would be the enzymes or the enzyme complexes for the electron transport chain. So the electron transport chain is obviously happening in here. So we have that over here. So cytochrome proteins are part of those enzyme complexes that contribute or that are part of the electron transport chain. The Krebs cycle happens in the matrix, so all the reactions for the Krebs cycle after we have acetyl-CoA that came from pyruvate, or other, it also comes from other places, I shouldn't just say from pyruvate, but pyruvate is one source of acetyl-CoA, and then we begin the Krebs cycle, and that happens in the matrix. We have the electron transport chain where we get the energy, and we're getting that energy from oxidative phosphorylation. Phosphorylation, let's do the easy part first. Not that the other part is hard. Oops. So phosphorylation just means adding phosphate. So say going from ADP plus a phosphate gives you ATP, which is energy. So that is, the main source of energy that we are making in most cells. Other cells will be phosphorylating other molecules, like say creatine in skeletal muscle plus phosphate gives you creatine phosphate, and that's an energetic molecule. Oops. So just adding a phosphate, you get energy. Now when we talk about oxidative phosphorylation, we're talking about redox reactions. Let's define that. Redox reactions are oxidation plus reduction. So we have oxidation reactions, we have reduction reactions. Oxidation reactions involves taking electrons. Reduction reactions involve hitting electrons. So these are paired because if you take electrons, you got to give them to something. And if you were giving electrons to something, you had to have gotten them from somewhere. So when you oxidize something, you're removing electrons away from it. When you reduce something, you are putting those electrons onto something, so a molecule. And that is what the electron transport chain is. It's just a bunch of redox reactions that end up with adding phosphate to ADP to create ATP. Let's just use NADH as our example to start in the electron transport chain. As we have NADH that we get from the Krebs cycle, we take electrons and let's just put one cytochrome protein here. So I'm going to get like a blob. So here comes an electron, just the negative charge, comes off of the NADH. We put it onto that cytochrome protein. So we oxidized NADH. We put it onto the cytochrome protein, so we reduced the cytochrome protein. Then we'll take the electron off of that cytochrome protein, put it on the next protein in the line. So we oxidize that cytochrome protein, reduce the next one, and so on and so forth as we go through the chain, which we're going to go into more detail in our next couple of videos. But just for now, just to understand that we are taking a substance that came out of the Krebs cycle, we're oxidizing it by taking the electrons away, we're using those electrons to reduce another substance, a protein in this case, and then we're going to oxidize that thing that we just reduced and use that electron to reduce another thing until finally, blah, 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 we do all that and we reduce an oxygen. So we are adding the electron to the oxygen in the end, and all those bouncing electrons create the energy to add the phosphate to ADP, and we get ATP. So that is what oxidative phosphorylation is, or phosphorylation is. So we are doing the Krebs cycle, we're doing oxidative phosphorylation in the form of the electron transport chain. We're also doing in the matrix beta oxidation. This is the fat breakdown, getting fats ready for the Krebs cycle, where we can do oxidative phosphorylation on them too. And then the last thing we need to know about the matrix is that the matrix has a high pH. It's about 8. So it's not super high, but it's higher than neutral. Most of the body is about neutral. So a little bit of a high pH. Okay, so that is it for the mitochondria. Our next video will be about ATP production and how we do it, or at least how we do it through glycolysis, the Krebs cycle, and the electron transport chain.